Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion and I'll ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speaker. So thank you for joining us today. It's now my pleasure to introduce our speaker, Sujeong Yang, PhD, Viral Vector Lead, Senior Research Scientist from Astrea Bioseparations.
Thank you for the nice introduction, Alex. I'm Sujeong Yang, Senior R&D Scientist working on viral vector applications in Astrea Bioseparations. Today, I'd like to present how we can minimize lentiviral vector loss during the purification process using an innovative nanofiber adsorbent. This is the outline of the talk. I'll start off with introduction of lentiviral vectors. Lentiviral vector contains a single-stranded RNA and is wrapped with envelope with protruding glycoproteins. It delivers a long-term and stable gene expression even to non-dividing cells such as neurons. These vectors manifest high complexity and sensitivity to low pH, high salt, temperature, shear forces, and so on. These characteristics make bioprocessing of lentiviral vectors very challenging in the field. It has pI value 6 to 6.5, so it will become negatively charged on physiological pH. This physicochemical property rationalizes the use of an ion exchanger for their purification. As advanced therapy medicinal product, lentiviral vectors are a very powerful tool. But its purification is challenging, especially achieving high yields of functional lentiviral vectors. On top of that, it is also challenging to set the scalable strategies for contaminant removal, such as host cell proteins and DNA. Here, I would like to introduce a novel intervention based on nanofiber technology, which can overcome those challenges. This is a novel composite electrospun nanofiber adsorbent called Astrea-Adapt. It has expansive flow path, allowing immediate access to the high surface area, giving a high binding capacity, fast flow, and low shear environment. This compresses downstream processing and allows handling of larger modalities used for cell and gene therapies as listed in the table here. This is the general workflow for LB purification from upstream to downstream processing. Cell culture expansion, harvest, clarification and filtration, and here conventional anion exchange-based purification done by existing platforms showing in the yellow colored box can be replaced by Astrea-Adapt nanofiber. This novel platform called Lentihero has been developed from the lab-scale spin column to the radial FPLC device for the largest scale production of lentiviral vectors. For the largest scale production of lentiviral vectors, suspension culture system is more commonly used than adherent system. However, suspension feedstock has significantly more host cell proteins than adherent culture feedstock, as you see the values in the table. Therefore, the clarification is a critical process for suspension LB feedstock prior to the purification. The aims of the study is to optimize clarification of suspension LB feed prior to the lentiviral capture by Lentihero adsorbent. Then to perform the lab-scale Lentihero spin column purification as a screening tool to find the conditions to maximize the lentiviral recovery and impurities removal. Finally, we can establish the purification workflow from the lab scale to Lentihero 1 FPLC device. We performed 3 clarification studies I will present data set from each study. This shows our lentiviral purification workflow using nanofiber Lentihero device. Depending on upstream, we vary the clarification process, mainly consists of primary clarification with or without buffer exchange. This can be performed by TFF or ultrafiltration. Alternatively, depth filtration can be used as a primary clarification. Then the clarified feeds were purified by Lentihero spin column as a recovery screening tool. This will help us to find the optimized conditions for maximum recovery and its purity. And we will finally move on to the Lentihero 1 FPLC device application. First of all, we aim to optimize buffer exchange volume for ultrafiltration step. And we performed buffer exchange with 1 time to 3 times buffer volume to the feed. Then we measured the infectious TU titer, HCP, and double-stranded DNA from the clarified feed. As shown in the graph here, 1-time buffer volume to the feed gave the highest TU titer with more than 40% of contaminant removal. When buffer exchange the volume increases, we lose more infectious titer. After the buffer exchange step, we did the recovery screening using the Lentihero spin column. As shown in the graph, 1-time buffer volume to the feed resulted in the highest TU titer with a 75% recovery to the load. Therefore, 1-time buffer volume to the feed was adopted for buffer exchange process in TFF or ultrafiltration to purify suspension LV with Lentihero FPLC device. Then we move on to the next study. In order to understand and reduce the shear stress from the start of the harvest, we explore the primary centrifugation from fresh harvest, followed by buffer exchange with ultrafiltration. For suspension feed, we tested different set of conditions with varied centrifugation speed and different molecular weight cutoff of ultrafiltration as you see in the table. Condition 3, which is centrifugal speed at 1500 G, followed by buffer exchange with 100 kilodalton filtration, gave the highest TU titer compared to the other conditions. It showed only minimal loss of infectious particles from the crude harvest. To assess the final recovery of purification, the Lentihero spin column was employed as a screening tool. It was operated parallelly, therefore, 6 conditions were screened within 25 minutes using the benchtop centrifuge. Condition 3 and 5 gave the highest TU titers for the final recovery. The only difference between condition 3 and 5 was the speed of centrifugation from the harvest. We chose condition 3, which is more gentle process with a slower centrifugal speed than condition 5. This means there will be less shear stress for condition 3. It was adopted in subsequent workflow. Finally, we move on to the comparison study. We directly compared 4 different clarification processes. Condition 1 to condition 4 covers standard centrifugation at 1500 G and test microfiltration with and without 100 kilodalton diafiltration and depth filtration with and without DE. Here you can see run details of each condition in the table. Infectious LV titers post-clarification was shown in the graph. Condition 1 to 3 showed more than 70% recovery, But the condition 2, which underwent 100 kilodalton diafiltration, gave the highest TU titer. Condition 4, which is depth filtration with DE, showed poor infectious titer among these conditions. It may be because of the positive charge on the DE binding lentiviral particles, which may affect the infectious titer adversely. Then the clarified feed went through the final recovery screening by Lentihero spin column. As shown in the graph on the left, condition 2 and 3 showed similar recovery, around 30% for infectious viral particle. As for the impurity level, condition 2 showed more than 95% removal for both HCP and double-stranded DNA, while condition 3 showed less removal of double-stranded DNA. Thus, condition 2 was adopted as the clarification for suspension feed prior to Lentihero purification. After we optimize the clarification process at the lab scale, at last we move on to the scale-up Lentihero 1 FPLC device. By diafiltration, we recovered nearly all lentiviral particles from the feedstock we tested. When the clarified LB feed was purified by the weak anion exchange fiber, we achieved a nice, sharp resolution for elution at the low conductivity. The final recovery of viral genome was nearly 70%. More importantly, functional recovery was 53% measured by Jurkat cells transduction assay. After we optimized the workflow using Lentihero 1 FPLC device, which is 1 mil nanofiber adsorbent, we achieved a high recovery and purity from both adherent and suspension systems. We processed 200 milliliter for suspension and 242 milliliter of adherent feed. Functional LV recovery was 53%, ranging from 2 to 510 to the 9 TU. Impurity level was also measured in the elution, shown in the graph. Around 98% of HCP was removed, and 82 to 85% of DNA was removed during total process. This graph shows scalability of LV yield from Lentihero spin column to Lentihero 1 FPLC device. The yield of Lentihero spin column was normalized to the fiber bed volume, which is purple bar in the graph. And this was directly compared to the recovery yield achieved by Lentihero 1 FPLC device. Strikingly, the lentiviral recovery yield from the FPLC device was exceeded over the expected yield as shown in the graph. This is a process modeling graph between feed volume and the required Lentihero nanofiber adsorbent. In order to process 50 liter bioreactor volume, which is clinical study scale, only 250 milliliter of nanofiber adsorbent will be required. Therefore, it can be predicted that 1 liter unit of nanofiber would process a 200 liter bioreactor in a single pass. This will satisfy the current demand for large-scale production of lentiviral vectors. This is the end of my talk. I would like to thank to our R&D team and management for supporting this work. Lastly, I want to highlight that we have 3 ranges of nanofiber chromatography devices for large-scale processes. You can see the names of the device in the slide. They are for purifying plasmid DNA, lentiviral vectors, and exosomes respectively. Thank you very much.
Okay, great. And as a reminder to our audience, you can go ahead and type in your questions for our speaker now. So our first question is, What are the unique properties of your Astrea Adept nanofiber compared to other fiber membranes?
Yes, our nanofiber composed of two different materials, one that gives strength and another for flexibility. So the effective size of the path through the fiber mat we made is much larger than other electrospun fibers. And our fiber is flexible compared to others, so it's possible to fit it into radial flow device, which can be easily scaled up.
And our next question is, what do you think are the critical parameters for the clarification optimization?
I think the most important factors to consider is reducing shear stress and processing time. It's because this will minimize the loss of infectious viral particles.
And why do you observe the increase in yield when you use the FPLC device for purification?
It's a very good question. Our Lentihero FPLC device contains a radially aligned electrospun nanofiber. So it's a radial device. So this radial structure makes the flow distribution more efficient. So the binding capacity and its viral recovery yield will increase compared to non-radial device.
All right, well, it looks like we're running a bit short on time, so if we don't get to your questions, we'll be passing them along to Sujeong, who can follow up with you directly. Please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, how long would it take to operate each Lentihero spin column as a screening tool?
The actual hands-on time for each run takes less than 25 minutes, but they are all run in parallel using the benchtop centrifugation. So we can run multiple columns at the same time, depending on the capacity of the rotor of your centrifuge in the lab.
Well, thank you, and thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.